Hello, this is Gina Piscatelli with a lecture for A&P 2 at Madison Area Technical College. In this lecture, we'll look at the last type of formed elements in blood, that's platelets, and their role in hemostasis, which is defined as the cessation of bleeding. Platelets have many functions in hemostasis, and one of the most important is coagulation. Coagulation is the formation of a blood clot. So first, let's just look at some information on platelets. Their Latin term is thrombocytes, and these are actually fragments of larger cells called megakaryocytes. Megakaryocytes fragment into these smaller platelets, which end up numbering 150,000 to 400,000 per microliter of blood, so lots, more than white blood cells. The formation of these thrombocytes is called thrombopoiesis, and like the formation of red blood cells and white blood cells, thrombopoiesis occurs in the red bone marrow. Thrombopoiesis is stimulated by a hormone called thrombopoietin, which is made in the liver. So after the platelets form, their functions are all related to hemostasis, or the cessation of bleeding. These platelets release chemicals, including serotonin, calcium, various enzymes, adenosine diphosphate, or ADP, and platelet-derived growth factor. Now, all these chemicals don't mean much right now, these names, but in total, they all function to help with hemostasis by causing vasoconstriction, which means the blood vessel will constrict, decreasing blood flow. So if there's damage to a blood vessel, you won't lose as much blood. Also, they function in the formation of what's called a platelet plug, which plugs a damaged vessel. And third, function in forming a clot or coagulation. So platelets, by releasing all these chemicals as well as physically binding together, perform all these functions: vasoconstriction, a platelet plug, and coagulation. First, we're going to go back to thrombopoiesis and talk about how platelets form. So just like we looked at erythropoiesis and leukopoiesis before, we start with a hematopoietic stem cell. So the hematopoietic stem cell is present in the bone marrow, and it differentiates eventually into a megakaryoblast. And then that megakaryoblast eventually becomes a megakaryocyte, or a megakaryocyte, excuse me. This megakaryocyte is big, that's why it's called mega, and it's in the bone marrow, and it extends these cytoplasmic and cell membrane, they call them tendrils or extensions, into a very large blood vessel that's permeating the bone marrow. We call this large blood vessel a blood sinusoid. So there's blood in this sinusoid that's flowing towards a vessel of the regular circulatory system. And as the blood flows past these tendrils or extensions, parts of the cell, these tendrils, break off. And so the platelets are formed by the breakage of parts of this megakaryocyte. Now, once the platelets form and enter the regular circulatory system, you know they release chemicals, but they're involved in 3 hemostatic processes that I mentioned before, all in order to maintain blood volume and prevent bleeding. Vascular spasm is one. The second is platelets form a plug at the site of damage. And lastly, they participate in forming a reinforced plug, I guess we call a clot, and that's coagulation. These 3 processes happen sequentially in response to blood loss and vessel injury. So first vasoconstriction occurs, which is the vascular spasm, and we'll talk about that in a little bit more detail, but the vessel becomes more narrow. Then the platelets aggregate and kind of form a wall where the damage occurred. This is called a platelet plug. And then finally a true clot forms. So they happen in sequence, 1, 2, and 3. So let's look at step 1, the vascular spasm. When there's vessel injury, the smooth muscle is damaged because the smooth muscle is present in the wall of the blood vessel. And when smooth muscle is damaged or pressed upon in any way, depressed, it automatically responds by constricting. So just the fact of damage does cause some vascular spasm, but it won't last very long. However, endothelial cells, those epithelial cells that line the inside of vessels, and platelets that are present in the blood, they both release serotonin when there's damage to a vessel. And this serotonin is continuously released because the damage occurs for a long period of time, of course, and that causes the smooth muscle contraction or the vascular spasm to last longer. So platelets prolong the vascular spasm that naturally occurs when smooth muscle is damaged. Now, also when a vessel is damaged, there's connective tissue around the vessel wall, and part of that connective tissue includes collagen fibers. And when the vessel is damaged or broken, collagen fibers are exposed. And platelets adhere to these exposed collagen fibers. So you can see here that they're adhering, even up at the top picture. When they do attach to the collagen fibers, 2 things happen, but collectively we call it platelet activation. So the first part of platelet activation is that they degranulate, meaning they've got granules that contain chemicals, but the granules release those chemicals, and those chemicals attract more platelets to the site of damage. The second thing that happens when platelets are activated is they become sticky because they become negatively charged. Now, platelet activation, as I mentioned, has these 2 events, the degranulation event and then the sticky event. So when degranulation occurs, this is what's released. These are the chemicals: ADP, serotonin, and thromboxane A2. ADP is what attracts more platelets to the site of damage. Serotonin, we know already, causes vasoconstriction, so prolonging the vascular spasm. And this thromboxane causes more activation of platelets. So that means that when platelets arrive, are attracted by this ADP, they will become activated by this thromboxane. And it turns out to be a positive feedback mechanism. As more platelets aggregate and become activated, then they too degranulate and attack attract even more platelets. Now, ultimately, when these platelets are activated due to damage, the membranes become negatively charged in response to degranulating. And that not only makes them sticky, but provides a surface for a plasma clotting factor to be activated. So we haven't talked about clotting factors yet, but clotting factors are proteins in an inactive form present in your plasma all the time. And they're activated by these platelets, the negative surface of the platelets, and so you'll get an activated clotting factor. So we'll come back to why that clotting factor is important in just a second. First, let's just talk about the degranulation event and this positive feedback mechanism that you have been introduced to. So there's damage first to a vessel. And we need that to be repaired eventually, of course, to completely stop bleeding. But the first thing that we notice happens besides the vascular spasm is that these are closed and platelets adhere to those collagen fibers, and they are activated when they adhere. And so then the platelets release all those chemicals we talked about. We'll just call them platelet factors. And ADP is one of them, which attracts more platelets, right? And they aggregate here, and the thromboxane will cause them to degranulate, and you'll get this positive feedback mechanism. So more and more platelets are attracted and activated through this mechanism. Now, the other thing that happens with platelet activation that I mentioned is that they become negatively charged once they're activated. So that's going to help with step 3, coagulation, because for coagulation to occur, we need that platelet plug to be reinforced and become a much stronger clot. And the reinforcement comes in the form of a protein called fibrin. So fibrin is a protein that you'll find in clots to make it stronger so that the platelets can't escape, and it provides a location for vessel repair as well. So the only way for fibrin to be formed is through a series of chemical reactions. And you don't want fibrin to be formed willy-nilly when there's no damage to a vessel, because we need blood flow to continue. So this coagulation, resulting in the formation of fibrin and then a clot, has to be very localized. It has to happen fast because you don't want to lose blood in the meantime. And it needs to be controlled so that it eventually stops. You don't want the whole vessel to form a complete clot from one side to the other. You need blood flow to continue. So we're going to look at the process, step 3 now, coagulation, the process of coagulation. So fibrin is formed by several chemical reactions that occur in sequence, like one reaction to second reaction, third, fourth, second, fifth, whatever. So we call this a cascade, and there's lots of reactions. We're not going to study every single one, but there's like 30 of reactions. Once this fibrin is formed, the platelet plug has been reinforced, but in addition, other blood cells adhere to the fibrin. So the clot is not only made up of the fibrin and the platelets, but also erythrocytes and leukocytes will, I should put an S on that. Leukocytes also become part of this clot. And in this picture of a clot, you can see the erythrocytes are red, the fibrin threads, the protein, kind of brown yellowish. And the leukocytes are green, and the platelets are like white-gray there. Okay, so clot has a lot of different components. But in order for it to happen, for coagulation to happen, formation of a clot to happen, we have to form this fibrin. Now, fibrin isn't always present in the plasma, but its precursor is. So there's a precursor to fibrin called Fibrinogen that's present in the plasma. And so Fibrinogen has to be activated. It has to become fibrin for a clot to form. And the way that happens is Fibrinogen is cleaved by another enzyme called thrombin to actually form fibrin. So if we just look at this picture for a second, which looks super confusing, but I'll try to break it down for you. The first thing is you've got some kind of damage to vessels. And then there's all these chemical reactions that take place. But ultimately, all these different chemical reactions involve activating a series of clotting factors that ultimately activate thrombin. And since thrombin is what's important for forming fibrin, you need thrombin to be activated. So multiple clotting factors will eventually result in a reaction that produces thrombin, which isn't always activated in the blood. It exists in the plasma as a precursor that's inactive called prothrombin. So a clotting factor will actually produce thrombin, and then thrombin will cleave fibrinogen, and you'll get this fibrin formation. So that's kind of just an overview. So in the plasma, we already have fibrinogen. In order to make fibrin, part of the clot, we need thrombin. And so thrombin has to be formed or activated by all these different clotting factors. Now, these clotting factors aren't constantly activated either. They exist in the plasma in an inactive form. And it's not until there's some sort of vessel damage will we see these clotting factors become activated. So the precursor, clotting factor 12, becomes activated. It will activate another clotting factor, which will activate another clotting factor on down the line until finally prothrombin turns into thrombin. And then we can get fibrin. Okay, so let's talk about clotting factors briefly. So these clotting factors, which I guess you could also include fibrinogen and prothrombin in this category, exist in the plasma all the time, but in an inactivated form. So we call them precursors or zymogens. Now they are enzymes in and of themselves. So they're not just proteins, they actually are proteins with an enzymatic function. They can catalyze a chemical reaction. And these zymogen clotting factors are made by the liver, and vitamin K is needed to produce them. So that's an important component of our diet. So how do we begin coagulation? Well, there's two pathways to start all these chemical reactions. There's an intrinsic pathway and an extrinsic pathway. And truthfully, they both happen simultaneously in conjunction with one another, but they involve different chemical reactions. So we're going to treat them as separate just to see why they're even called something different. So first you have damage to a vessel wall, right? And in the intrinsic pathway, clotting factors are going to be activated due to the activation of platelets, which are present in the blood. So the intrinsic pathway will start coagulation by the platelets. The platelets will begin the intrinsic pathway. And it's called intrinsic because platelets are in the blood. So intrinsic to the blood. Versus the extrinsic pathway. So extrinsic pathway, the cells of the blood vessel wall will also initiate coagulation through a series of chemical reactions, but it starts by the release of a tissue factor. So tissue factor is released into the blood, and that's going to start a process of coagulation as well. So if we go back to these different steps that we originally looked at to form fibrin, which is our component of a clot that we really need, we've got two ways that it begins. Intrinsic pathway and the extrinsic pathway. In the intrinsic pathway, platelets that have been activated also activate clotting factor 12. That's the beginning of that whole pathway. In the extrinsic pathway, the vessel wall that's been damaged, not a blood component, but the wall of the vessel, releases a chemical known as tissue factor. And that will activate a different clotting factor. I don't care that you remember it, but it's number 7. Okay. So once a clotting factor has been activated, whether it be 7 or 12, you're going to get a series of chemical reactions that ultimately will activate a zymogen called Prothrombin Activator. It's another clotting factor. Doesn't matter. Now notice in this first step of coagulation or fibrin formation, regardless of how we start, either the platelet started it or the damaged tissue started it, you're going to need calcium at some steps. So calcium is a requirement for clot formation, another important role for dietary calcium. And the end of this first step is to form this thing called Prothrombin Activator. Now what that means is that chemical will activate prothrombin. Now prothrombin is a precursor or a zymogen that's in an inactive form, and so Prothrombin Activator will activate prothrombin to become thrombin. So that's step 2. Now we have thrombin at the end of step 2. That's what we need to cleave fibrinogen into fibrin, which again is the component of a clot. That's the main component of the clot that we need to reinforce a platelet plug. Fibrin will cross-link and form this intricate meshwork with the help of some other clotting factors and calcium. So what I think is important for you to remember is that there's 2 pathways for initiating coagulation, the intrinsic pathway and the extrinsic pathway. The intrinsic pathway involves platelets activating a clotting factor. The extrinsic pathway involves the damaged tissue releasing tissue factor. Both of those will result in the formation of Prothrombin Activator. Then I think you need to know that Prothrombin Activator forms thrombin. And then I think you need to know that thrombin is an enzyme that turns fibrinogen into fibrin. And finally, I think it's important for you to remember that calcium is needed at various points. You don't have to know exactly where, but calcium is required for all these chemical reactions to occur completely from the beginning to the end, step 3, and that a lot of these clotting factors and enzymes are present in the plasma in inactive forms and aren't activated until there's damage to a vessel. So this is the intrinsic pathway in a little bit more detail. If you really need this to make sense of things. So that's step 1 from initiation down to formation of Prothrombin Activator involves many different clotting factors and starts with platelets activating the first one. So here's the inactive form of clotting factor 12. And then 11 is used, and 9 is used, and then 8 is used, and then 10 is used, and then 5 is used. You don't have to remember all that. But notice that once 12 has been activated, it converts inactive 11 to active 11. And then activated 11 converts inactive 9 to clotting factor 9 activated, and whatever, on down the line. Okay, so 12, 11, 9, 8, 10, and 5. But you don't have to know that. The extrinsic pathway, instead, for that first from initiation down to formation of Prothrombin Activator, starts with the damaged blood vessel releasing tissue factor. And again, you're going to have a cascade of reactions that ultimately produce activated clotting factors. But you start with tissue factor, which converts inactive 7 to active 7. Activated clotting factor 7 converts 10 to activated 10, and then to 5. Eventually, you get this thing called Prothrombin Activator. So we can move on to step 2, which is the formation of thrombin. But anyway, so comparing the extrinsic versus the intrinsic versus the extrinsic initiation of coagulation. Intrinsic means that coagulation is initiated by substances present in the blood. That means platelets and clotting factor 12 inactive. It's slower. The extrinsic pathway is initiated by substances outside of the blood. That means the blood vessel wall. Damaged tissues release tissue factor. This is faster. I don't know exactly why. It's got fewer chemists, maybe. Both pathways involve activating clotting factors, and both pathways end with, activating this prothrombin activator, and you're going to need calcium at some point. So that's a summary of step 1 of coagulation. Step 2 and step 3 are so easy in comparison. Once you have prothrombin activator, that just means you're going to convert prothrombin into thrombin. It activates prothrombin, and you get thrombin. So the end of step 2 is thrombin formation. And then in step 3, thrombin will convert fibrinogen into fibrin. So you get the formation of fibrin and this cross-linked meshwork. So that's coagulation. But, and platelets start, that they happen at the same time, and platelets are involved in fibrin formation. But they have more functions than just coagulation. They are involved with helping the clot to retract, because we need the platelets to initiate the formation of the clot, but we then need the clot to compact, become kind of tighter and smaller, to bring the edges of the blood vessel closer together so that repair can happen. So the clot begins to retract in response to platelets, some chemical that the platelets release. In addition, the platelets release this thing called platelet-derived growth factor, PDGF, and that stimulates cells in the vessel wall to undergo mitosis, divide, healthy cells. So smooth muscle cells and fibroblasts will divide, and that will rebuild the vessel wall. So the platelets formed the clot, they retract the clot, and they participate in vessel repair, getting healthy vessel wall tissue to form. In addition, the clot does need to be dissolved. You don't want it to stay there. Even, the vessel wall is fine. Now we can get rid of the clot to keep blood flow moving. And that's only going to happen if a certain enzyme is activated, and the enzyme is called plasmin. It breaks down the fibrin. And so everything will sort of dissolve and break up into inactive forms. So by activating clotting factor 12, platelets contribute to the activation of clot breakdown. Clot breakdown is called fibrinolysis because the plasmin lyses the fibrin. So clotting factor 12, once it's activated, will also begin activating plasmin. And that'll start, it's slower than the formation of the clot, but eventually plasmin will be needed to break down the clot. So those platelets have a lot of function. Not just the formation, but the breakdown of the clot as well. Okay, now let's talk a little bit about abnormal coagulation and terminology related to it. So the process of forming a clot in an abnormal situation where the vessel is not broken is called thrombosis. And the clot itself is called a thrombus. And this most often occurs in leg veins due to inactivity or immobilization. And that's just because of the accumulation of even inactive clotting factors and blood pooling, and so a lot of platelets get there. And some of these chemical reactions just happen spontaneously. And over time, if you don't keep the blood flowing, there's a buildup of these factors that can form a clot. Now, it is possible for the thrombus or the clot to not stay stationary, instead float or travel once blood flow, kind of picks up a little bit. And so a traveling thrombus we call an embolus. And if it does reach this embolus, if it does reach a narrow vessel where it obstructs blood flow, we call it an embolism. And some of you might have heard of a pulmonary embolism, which is a clot that travels from veins to really small vessels, capillaries, in the lungs. So it's called a pulmonary embolism, which is bad because if the blood is not flowing through the lung tissue, it's not picking up oxygen. So that's bad, right? Let's talk a little bit about a few other terms. Like infarction, ischemia, and stroke. So an infarction is actually tissue necrosis or death. So you've probably heard of a myocardial infarction. That's where part of the heart muscle dies. It's damaged. Now, why would it happen? It can happen for a variety of reasons, but one reason could be due to the lack of blood supply. Oxygenated blood, that is, because that's what the problem is. The lack of blood supply is called ischemia, and of course that could happen due to a clot forming in a vessel surrounding the heart wall. So a clot could result in tissue death. When this kind of event occurs in the brain, we call it a stroke. So you have ischemia in the brain. resulting in tissue necrosis or death. Part of the brain isn't going to function anymore. That's referred to as a stroke. Now, the last term I wanted to point out is a hematoma. A hematoma is a mass of blood that has left blood vessels and gone out into the tissue. Usually it's clotted. Okay, so it's like kind of goopy, whatever. A clot we normally think of as forming within a vessel. But in some cases where you've had a lot of blood loss, this blood that has left the vessels and gone out into the tissues can also clot. That's called a hematoma. Now, since we have all these clotting factors present in the plasma, although in inactive form, there are some things the body has to prevent unwanted clots, because the clotting factors, as I mentioned, could spontaneously become activated. So one thing that helps the prevention of clotting is that the blood is flowing rapidly, and that dilutes the clotting factors as well as, prothrombin and everything. So that prevents the buildup of critical levels at a certain location. That's good. In addition, the vessel wall, all of our vessels, release chemicals that prevent platelets from adhering, just spontaneously or randomly adhering. And the vessel wall chemicals that do this are called prostacyclin and nitric oxide. So it just won't help. It won't promote platelet aggregation at a certain site. Obviously, when there's damage to a vessel, the prostacyclin and the nitric oxide aren't enough to keep the platelets from adhering. And then in addition, we have white blood cells that interfere with the formation of thrombin, and these white blood cells are basophils. They release heparin, and that heparin actually interferes with that very first step, formation of Prothrombin Activator. But it helps prevent unwanted clots. And although the liver makes clotting factors, it also makes antithrombin at a certain level. And antithrombin deactivates thrombin. And it's just a fine-tuned balance. It's not like antithrombin will overcome all thrombin action. It's just a balance so that any thrombin that spontaneously forms will probably be in a small volume, and there'll be enough antithrombin present to deactivate it. However, when the coagulation events begin from initiation on down, there'll be so much thrombin activated that the antithrombin quantity won't be enough to overcome the formation of a clot. I hope that made sense to you. Now, clinically, we manage blood clotting in different ways. In one way, we do it by preventing clots from forming at all. And in another way, we dissolve clots that have already formed. So let's go with prevention first. There are certain medicines called anticoagulants, coumarin, warfarin. These are all vitamin K antagonists so that the clotting factors can't be made. And aspirin suppresses thromboxane, so that will prevent platelets from becoming activated, so you won't get clots forming willy-nilly without control. And leeches actually make an anticoagulant that inhibits thrombin formation. I don't know if they still use that much or not anymore. They might. Okay, dissolving clots that have already formed, we also have a way to deal with those. streptokinase is an enzyme made by a bacterium and that can be applied to dissolve clots in coronary vessels. And then there's this thing called tissue plasminogen activator that just increases the amount of plasmin present in the blood, which, if you remember, plasmin is what breaks down a clot. Don't memorize all those. Okay, there's a few coagulation disorders that we should talk about and platelet disorders. So some disorders that would impair coagulation would be any kind of liver disease, because the clotting factors would be, or synthesis of the clotting factors would be impaired. And then there's a set of disorders, coagulation disorders, referred to as hemophilia. And you might have heard of these as a bleeding disease. These are hereditary diseases, so that means there's a genetic component. And the genetic component will be related to a gene that codes for clotting factors. So there will be certain genes that are mutated, so you won't get a clotting factor. There will be clotting factor deficiencies. There's a lot of different hemophilias. One is sex-linked hemophilia. That means that the gene is on the X chromosome, and so factor 8 or factor 9 isn't made, inactively or actively. It's just not present. And then there's autosomal hemophilia. And autosomal means that it's not on the X chromosome. There's a gene on chromosome 4 that codes for factor 11, but the gene is mutated, so that factor's not present. And then there's another coagulation disorder, von Willebrand disease. I don't know where the gene is located, but there's probably variants. But in this case, platelets won't adhere to a damaged vessel correctly. They don't respond to the presence of those collagen fibers. Okay, and the last bit of disorders related to platelets would be the thrombocytopenia. So that means, penia means reduced, so you have reduced thrombocytes. And this is defined as a platelet count below 50,000 platelets per microliter. And it's usually caused by bone marrow destruction. It could be due to the presence of a cancer like a leukemia, but more often than not, it's due to a treatment that was given to the bone marrow, like radiation or chemotherapy, and now platelets don't form as often or as much. And signs and symptoms would be spontaneous bleeding from small or large blood vessels, but we'll go with small because thrombocytopenia, you still have some platelets there, so you can get normal clotting. But what happens is normal movements, little bumps, lead to widespread, very small hemorrhages in blood vessels. And these small hemorrhages can be seen on the skin. There's different categories of these hemorrhages. If they're really small, we call them petechiae. If they're a little bit larger, we call them purpura. But that might indicate a low platelet count. Okay, that's the end of our coagulation lecture. Thank you very much. And platelet lecture.